Now hi guys, welcome back to a fresh new video. So in this video we are going to discuss the important MCQ of the chapter biological classification. So see, it is one of the most important chapter in the class 11 biology. And what happens in this chapter, if we see the modules and the other references book, then you will find the different things related to these chapters and which are irrelevant and not so important. But the things that given in NCERT is very important. So A to Z NCERT is important line by line, word to word. So let's see. So first of all, see, the all organisms in the earth are differentiated into different kingdom, right? So first of all, in the lineage time, lineage means in Carolus lineage time, there was a two kingdom classification, that is kingdom Plantae and the kingdom Animalia. All the organisms are classified under kingdom Plantae and Animalia. Those who have a cell wall are classified in the kingdom Plantae, and those who have not a cell wall are classified in the kingdom Animalia. And it was very simple, but this is not sufficient, right? There are so many organisms which can be classified under kingdom Plantae and Animalia. So at later a third kingdom classification is discovered, and the new kingdom that added to the kingdom Plantae and Animalia is the kingdom Protista, and it was given by Haeckel, remember. So the third kingdom added is the kingdom Protista. So later fourth kingdom classification came, and at last in 1969 R.H. Whittaker gave the five kingdom classification. So this is R.H. Whittaker in 1969 proposed the five kingdom classification, and these kingdom are Monera, Protista, Fungi, Plantae and Animalia. Remember the scientist name and also the year 1969. The main criteria for classification used by him include cell structure, body organization, mode of nutrition, reproduction and phylogenetic relationship. Remember every word: cell structure, body organization, mode of nutrition, reproduction and phylogenetic relationship. Important MCQ, right? Now see, this is one of the most important chart. Here the characteristics of the five kingdom classification. So remember the whole chart, but the most important things are the cell wall composition. In case of Monera, the cell wall is non-cellulosic. In plant, the cell wall is cellulosic. In Monera, this is non-cellulosic, and that is formed by polysaccharide and amino acid. Remember, polysaccharide and amino acid. In fungi, it is formed by the chitin. One important thing about chitin is a polymer of N-acetyl glucosamine. Remember, chitin is a polymer of N-acetyl glucosamine. So see, in Plantae, the cell wall is made up of cellulose. Now, in the mode of nutrition, the important is in Animalia, the mode of nutrition is heterotrophic, and it is holozoic and saprophytic. Remember, heterotrophic, and in case of heterotrophic, it is holozoic and saprophytic. Important. This whole chart is important. Now see this. This is a controversial question. To see how. Here, kingdom Protista has brought together Chlamydomonas chlorella, earlier placed in algae within plant and both having cell wall. To say Chlamydomonas and chlorella, as you find in the plant kingdom, these two are included in the kingdom Plantae and under the class algae, right? But here it is written as a kingdom Protista has brought together Chlamydomonas and chlorella. So you have to also remember this line. So the question may asked as a Chlamydomonas and chlorella are brought together by the kingdom, then answer will be the kingdom Protista. Remember, it is a controversial question. Now see the phylogenetics. The word phylogenetics is important, and phylogenetics means it is based on evolutionary relationship. So remember the word phylogenetic. Now see 2.1 kingdom Monera. We will be discuss the kingdom one by one. So first kingdom Monera. So see, bacteria are the sole member of the kingdom Monera. Remember, in kingdom Monera, only member is the bacteria. Only bacteria is present in kingdom Monera. So see, bacteria are grouped under four categories based on their shape. Based on their shape, the bacteria are classified under four categories. These are spherical coccus, these, then the rod-shaped bacillus, these, then the comma-shaped fibrium, these, and the spiral-shaped spirillum, this. I have also made a video of important diagram of this chapter, so you can visit this. But here we will generally discuss the important MCQ. So remember the shape and the example. Now here, most extensive metabolic diversity is present in the kingdom Monera. So remember, in kingdom Monera, most extensive metabolic diversity is present. Now see, in the kingdom Monera, again can be classified into the Archaebacteria and the Eubacteria, also known as true bacteria, and the Archaebacteria, also known as ancient bacteria. Right? Now see, the Archaebacteria are again classified into the Halophiles, then the Thermoacidophiles, and the Methanogenous. Right? Halophiles are present in salty areas, Thermoacidophiles in hot spring areas, and the Methanogenous are present in marshy areas. To remember these three words. Archaebacteria differs from other bacteria. Other bacteria mean from true bacteria in having a different cell wall structure, and this feature is responsible for their survival in extreme conditions. So the Archaebacteria are different from the Eubacteria, that is true bacteria, in having a different cell wall structure. Remember, important MCQ. Different cell wall structure. So Archaebacteria have a different cell wall structure, and for this structure, they are survival in extreme condition. Important. Methanogenous are present in the gut of several ruminant animals, such as cows, buffaloes, and they are responsible for production of methane, that is biogas, from the dung of these animals. Important. Now let's discuss Eubacteria. There are different types of Eubacteria. One is a Cyanobacteria. Cyanobacteria have a chlorophyll A similar to the green plant. Remember, in Cyanobacteria they have a chlorophyll A that is present in plants. It is similar to that of plants. And also Cyanobacteria are photosynthetic autotroph. Remember, Cyanobacteria are photosynthetic autotroph. They are only member in kingdom Monera. Those are photosynthetic autotroph. Now heterocyst. These are the heterocysts. Heterocysts are the nitrogen fixing organ in Cyanobacteria. Remember, nostoc and anabena, classic example of Cyanobacteria. Chemosynthetic autotrophic bacteria oxidize various inorganic substances such as nitrate, and ammonia. Now see, they play a great role in the recycling nutrient like, remember the nutrient nitrogen, phosphorus, iron, and sulfur. So in question, they can give this for nitrogen, phosphorus, iron, sulfur, and one another, for example calcium, and will be asked what is the element which is not recycled by the true bacteria. So then answer will be the calcium, because nitrogen, phosphorus, iron, and sulfur are recycled by the true bacteria. So remember these four elements. Now see, heterotrophic bacteria are the most abundant in the nature. So in the nature, among all these bacteria, that is photosynthetic autotroph, chemosynthetic autotroph, the heterotrophic bacteria are most extensive, are most abundant. Remember. Now see, the citrus canker. Citrus canker is caused by a bacteria that is a Xanthomonas. Remember the name also. Now mycoplasma are organisms that completely lack a cell wall. So mycoplasma lack a cell wall, and the mycoplasma are also known as PPLO, that is pseudo-pneumonia-like organisms. PPLO. Now mycoplasma can live without oxygen. Important. Mycoplasma can survive without oxygen, and the mycoplasma are pathogenic in both animal and plant. Remember, and the mycoplasma are pathogenic in case of both animal and plant, and that they can survive without oxygen. Remember. Now 2.2, kingdom Protista. That is kingdom Monera is finished. Now kingdom Protista. Now kingdom Protista, what happens in kingdom Monera? The bacteria are present. That is bacteria are prokaryotic, right? Now in kingdom Protista, they are all single-celled eukaryotes. In Protista, all eukaryotes that are single-celled are present. And in kingdom Protista, they do not have a well-defined boundary, right? Now see, Protista reproduced bisexually and asexually both, and by a process involving cell fusion and zygote formation. Remember the two words, cell fusion and zygote formation. Important MCQ. So there are different types of Protista. The first class is Chrysophytes. Right? Now Chrysophytes are two types. There may be diatoms or golden algae, also known as desmids. Remember, diatoms and golden algae. Golden algae are also known as desmids. Now Chrysophytes may present in fresh water as well as marine environment also. That means they are freshwater as well as salty water animals, right? Now the plankton. What is the plankton? Microscopic and floats passively in water current. Remember, planktons are microscopic and passively floats in water current. These are plankton, remember. Now diatomaceous earth. Here the process of formation of diatomaceous earth is given. It is important for board, and the diatomaceous earth is important for NEET also. And the diatoms are also known as chief producer in ocean. Diatoms are also known as chief producer in ocean, remember. Now second class is the dinoflagellates. Now important about dinoflagellates are they may be yellow, green, brown, blue or red depending on the main pigment present on their cell. They may be found in various colors: yellow, green, brown, blue or red. Okay. Most of them have two flagella. Dinoflagellates have two flagella. This is dinoflagellates. They have two flagella. One lies longitudinally and the other transversely in a furrow between the wall plates. So remember this. Most of the time two flagella, right? One lies longitudinally and the other transversely in a furrow between the wall plates. Now example of dinoflagellates is goniolax. And one important thing about goniolax is that they form red touch. They form red touch. And the red touch is nothing but the red dinoflagellates, that is goniolax. When deposited on the water surface, they become red in color, and this is known as a red touch. Now toxin released by such large number may even kill other marine animals and fishes. So the dinoflagellates are toxic in nature. Now another class is Euglenage, right? This is Euglenage. Now Euglenages are freshwater organisms. To say the diatoms that are chrysophages, these are both marine and freshwater. Dinoflagellates are exclusively marine, and the Euglenoid are freshwater organisms. They are found in stagnant water. Stagnant water means, for example, this is a hole in the air water deposit, and this is known as stagnant water. Here Euglenoids are present. So they are found in stagnant water. Instead of cell wall, in Euglenoid they don't have a cell wall, but they have a protein layer called pellicle. This layer is known as pellicle, right? And it is flexible. Due to proteinous nature, it is flexible. Euglenoids also have two flagella, one and two flagella. One is long and the second one is short. Though they are photosynthetic in presence of sunlight, when deprived of sunlight, they behave like heterotroph by predating on other small organisms. So remember, in case of Euglenoid, they have a chlorophyll A, as similar to the plant, right? Cyanobacteria also have chlorophyll A, similar to the plant, and Euglenoid also have chlorophyll A, similar to the plant. But in absence of sunlight, when they can't produce food through photosynthesis, then they act as a predator and they predate the smaller organisms. So remember, they are also heterotroph and a photosynthetic autotroph. Interestingly, the pigment on Euglenoid are identical to those present in the higher plant. So I have said already, they have chlorophyll A. Example of Euglenoid is Euglena. Remember, this is diagram is also important. Now slime mold. The important thing about slime mold, this is slime mold. Slime mold is they have naked cytoplasm. Remember the word because it has already asked in previous question. Slime mold are a saprophytic protista. They are saprophytic protista. Another important thing about a slime mold is that they form plasmodium in a suitable condition, and during unfavorable condition, the plasmodium get differentiated and they form fruiting bodies bearing spores and their tips. So in suitable condition, they form plasmodium, and during unfavorable condition, the plasmodium get differentiate and form the fruiting bodies. And the spores of the slime mold have a through cell wall. For example, this is spore, then this will be the through cell wall, right? And they are very hard, that they are undestructible, and the spores are dispersed by air currents. These spores have true cell wall and extremely resistant and survive for many years. And these spores are dispersed by the air current. Important. Now protozoans. Protozoans are again differentiated into the amoeboid protozoa, then the flagellated protozoa, ciliated protozoa, and the sporozoans. Now, in case of flagellated protozoans, the example is trypanosoma, and the sleeping sickness, that also known as African sleeping sickness, happens due to the trypanosoma, and the parasitic form of this causes disease such as sleeping sickness. So flagellated protozoa and disease causing, And trypanosoma causes the sleeping sickness, also known as African sleeping sickness. Now, ciliated protozoa. The ciliated protozoa example is the Paramecium. One important thing about Paramecium is they have a dimorphic nucleus, one macronucleus and one macronucleus. Paramecium have dimorphic nucleus. Important. Ciliated protozoa. So see, flagellated protozoa mean, for example, this is protozoa, right? This will be the flagella, flagellated protozoa. Then ciliated protozoa mean, for example, this is the protozoa. Then this will be the cilia, hundreds of cilia, hundreds of thousands of cilia. Now sporozoans. Sporozoans are nothing but the maral parasite, that is a Plasmodium 5x, etc.